Hello, my name is Genevieve Parkey, and I'm going to briefly talk to you about my project this summer that I did at Lawrence Livermore National Laboratory, system-level analysis of metabolism in an algicidal bacterium. To answer many of the LLNL's mission-related biological questions, we conduct system-level analyses of natural ecosystems. During these investigations, we often discover new bacteria which lead to many questions. One important question that needs to be answered is, what do each one of the newly discovered bacteria need to survive? This summer, my team and I have been working to answer this question for a newly identified bacterium called Rickettsia-like diatom killer, or RLDK. This is a bacterium that attaches to Phaeodactylum tricornitum, a model microalga that can be harvested for biofuel, and it destroys it for reasons still unknown to us. This is an important study because as climate change continues to be a huge problem, we are trying to get more of our energy needs met by biofuels. If there's a killer bacterium lurking, understanding the interaction that it has with algae will help us maintain levels of biofuel production in the future. Using the metagenome assembled genome of this bacterium, we annotated it with 4 different bioinformatic tools. In other words, we identified all the genes in the genome of the bacterium and assigned biochemical functions to the proteins that are translated from each gene. We imported the annotated genomes into the U.S. Department of Energy's KBase platform and used the available tools to build automatically generated draft genome-scale metabolic models of the organism according to each annotated genome. These models correlate genes with different pathways and reactions present in the organism and provide insight into the metabolism of it, and might even give clues to why it's killing the algae. For now, we have been focusing on determining essential exchange reactions from these models. This will inform us of the metabolic capabilities and needs of the bacterium. It will tell us what nutrients it can make and also what it must import for survival. After comparing the features of the different models, we decided that the model generated based on the annotation from RAST seems to be the most complete, with the highest number of gene function assignments, and therefore it could be the most reliable for examining the metabolism of the killer. Using CobraPy, a genome-scale modeling package for Python, we produced lists of the essential exchange reactions based on the metabolic models. Taking that list, I have been examining the pathways that correspond to these reactions, and whether or not they're operational in the bacterium. So far, we have found that it might be able to produce histidine, an amino acid which our experimental colleagues have shown can regulate its algicidal activity. Our initial hypothesis had been that perhaps the bacterium kills the algae for this amino acid, but the results seem to disprove that hypothesis. Still, discovering that this bacterium can produce these metabolites is just the beginning of understanding this organism. Using the metabolic models, we aim to uncover other critical metabolites that can and cannot be produced by it, and more research will also give us insight into the role of possible transporters in its membrane, as well as other features that will help us determine why this organism is killing algae and causing crashes in ponds where it is grown and harvested for biofuel production on an industrial scale. And soon, we will be one step closer to helping produce cleaner energy for our planet. Thank you for your time.